Guys, thank you very much for joining our class. Um, let's talk about the memorandum of or suggested answers of this paper you wrote. I know that some of you smashed this paper, most especially if you were among the people who were following us. Yeah, I hope you managed to score high. And then don't be challenged by anything. For example, maybe my answer is a little bit different from your answer. Yes, it could be, but it could be different answers. I cannot discuss each and every answer which is most likely to be in this paper. So, guys, let's start without wasting time. Our paper, so that we are able to get it right. I thank everyone who shared, who were on this discussion of the prediction, the late revision, everything. Thank you very much. May God reward you, and we pray that you have a successful life. If things don't go the way you expect them to go, please still don't lose hope. If one door closes, obviously there will be another door opening for you. Just keep opening the doors. You'll be able to get one which is right. All right. 1.1.1, we are saying that which female scientist took X-ray diffraction X-ray of DNA molecule and concluded that DNA is helical shape or helix shaped. In this case, all of them, these people, all of them were among the people who discovered the structure of DNA. They just talked about one name. James Watson is a man. Francis Crick, they were working together, is also a man. Rosalind Franklin is also a man. So the answer is, did I say Rosalind Franklin? Maurice Wilkin is a man, and then Rosalind Franklin is a lady. Is the lady who died of cancer. Yes, so the answer, the most correct answer in this case is supposed to be B. A cell with the 14 chromosomes undergo meiosis. In this case, they're saying a cell with 14 chromosomes. They didn't say that this is a diploid, homologous chromosome. They just a cell with 14 chromosomes. They didn't say that a cell with homologous chromosome. So it means that they're just talking about 14 chromosomes. If they're homologous, then automatically it means that there are 7 chromosomes. So a cell with 14 chromosomes undergo meiosis. What is the number of chromatid? They're asking for the chromatid. Remember, each chromosome is having 2 chromatids. So they're asking what is the number of chromatid in this cell at the beginning of meiosis. So they didn't say homologous, therefore it is 14 times 2, so that it gives you 28. I know that some people wrote you are 58, but they didn't say homologous. They said just chromosome. You understand? If they had said homologous, then you would have said 56, but they just said 14 chromosomes, which means that it's just a single chromosome. Yes. So the diploid of it, meaning that it is 7. Yes. All right. So the most correct answer becomes C, which makes it to be the most correct answer in this regard. They're saying that inheritance of blood group are two. Remember when we are talking about on the prediction about the blood group, I said that blood group is controlled by one gene. Yes, one gene, 3 alleles. Yes, 3 alleles, and then we say that four phenotypes, phenotypes, and 6 genotypes. 6 genotypes. One gene, that's gene, just a gene, and then gene for blood group. 3 alleles, allele for A, allele for B, and allele for O, and then 4 phenotypes, that is A, B, A, B, A, B, A, B, and O. And then 6 genotypes, we saw them. So we come here, we say that 2 phenotypes, that one goes out. 3 phenotypes, we said there are 4 phenotypes, that one goes out. 4 phenotypes correct and 3 alleles. You see 3 alleles, so this one becomes the most correct answer. 4 phenotypes and 2 alleles. No, it is 3 alleles. So the most correct answer in this regard becomes C. So you get 2 marks there. Yes, guys, don't panic because you're not getting it right. Yeah, there are other questions where you're gonna get them right and still you stand to score a distinction. Yes. The following statement relates to cell division. Okay, crossing over, they're saying contributes to genetic variation in the population. Okay, they're saying that produce cells with the haploid number of chromosomes produce somatic cell. Haploid, diploid number chromosome cells with the diploid number, this is very rare unless it's fertilization. Somatic cells only, not really. So okay, well, let's see what they're saying in this regard. They're saying that which one of the following combination of statements refer to meiosis? Meiosis. Obviously, meiosis result in haploid, so not diploid. So this one, whenever there is 3, is off. So this is off. This is off. So it's off. So expect to have this and this. They're saying that produce somatic cells. Meiosis doesn't produce somatic cell. Meiosis produces gametes. So therefore, anything which has 4 is out. So automatically we only remain with B. So B becomes the most correct answer. Then we go to 1.1.5. They're saying that op, some and beds are believed to have originated from common ancestor. The shaded part of the diagram below shows the distribution of these species. So you have these shaded parts. So the distribution of the species. So in this case, what are we looking at? The type of evidence for evolution. The type of evidence for evolution that is represented in the diagram is, in this case, we are seeing that different lives are there. So therefore it is bio, because the different geography of the world therefore becomes biogeography. So A becomes the most correct answer. They're saying that the plant with yellow which is recessive, round which is dominant, is crossed with the green which is capital letter which is dominant, with wrinkled which is recessive. So meaning that you have capital Y, small y, and then capital R, small r, capital R, small r. So in this case, what are they asking? They're asking that which one of the following are possible genotypes of the parents one. So which parents resulted in the formation of this? So definitely I will need to have one which has a capital letter and one which has a small letter. Yes, so that when they combine, they form this, and then this. So if you look at this one goes out, not really, because of yes, this one, it goes out also. Yes, this one goes out. So the most correct answer, because this one will come, bring it this one there, and then this one brings that there, then this one brings that there. Yes, and then this brings that there. So the most correct answer becomes A. Yes, so that is it. We go to the next two. If this is 12, definitely this is 12. They're saying 27% of guanine is strand B. If this is 27, also this is 27. All right, because they are complementary to each other. So now they're asking you, they're saying that uh, they're saying that in the segment of DNA, there will be also 31% of adenine in strand 2. If you look at strand 2, 31% of adenine doesn't look to be right. 31% of adenine. If you look at this strand 2, 31% of adenine. Adenine, no, it's not indicated in this case. So that one's out. 12% of cytosine in strand 1, 12% of cytosine, we said is 27. So that one's also out. 31% of thymine in strand 2. Thymine in strand 2 is 31. Let's see the last one, 27% of cytosine in strand 2. Cytosine in strand 2 is 12. So definitely the most correct answer of meiosis, where chromatids are put opposite. They are saying chromatid. They are not saying chromosomes. They are saying chromatids. So in this case, when we talk about chromatids, it must be in a meiosis II. So therefore, where there is meiosis I is out. So the answer is either this or this. But they're saying chromatids. If they are chromatids, it means the chromatids are the one which are being pulled to opposite poles. This happens in anaphase. So in anaphase, if that's the anaphase, then automatically it's anaphase II. So D becomes the most correct answer. Yes. Huh. Then we go to the next question. They are saying that the next correct question, they are saying that the section of DNA molecule that code for a specific character, the section of DNA that code for a specific character is called a gene. Is called a what? A gene. Then we go to the pattern of black bears resulting in DNA analysis. We call it DNA profile. Not DNA profiling, is DNA profile. So punctuated equilibrium. Yes, it's punctuated equilibrium. Then we go to the next, the production of genetically identical copy of organisms using biotechnology. We call it cloning. Cloning, yes. Call it what? Cloning. Then they are saying that undifferentiated cell in animal that have the potential to become any type of tissue. How do you call that undifferentiated cell? The undifferentiated cell, we call them stem cells. Stem cells. We call them stem cells. Then we are saying that a genetic disorder, genetic disorder that result in a person who cannot distinguish between colors. We call it color blindness. Color, color blind, color blindness. Yes. The part of the plant where male gametes are produced. The part of the, I don't want to go to this like booklet distinction material to show you. Where are you? Where are you? Yes, must be around here. You see, we call them what? You see, even emphasized about it. You see that this is the point that you call them anthers, and then they say the male. If you check here on the question, they say that the male gametes. So we see here that the male gametes are produced in plants, are produced from the anthers. And how do you call those male gametes? We call them pollen grains. Yes. So guys, that's what is supposed to. Yes. Then we go to the next question. The next question, they're asking you that the structure that join two chromatids together. How do you call the structure that joins two chromatids together? We call them the centromere. Centromere. So we call it a centromere. So you see, you have got 8 out of 8 in that regard. And then I think that guys, you are achieving to that point. Then let's still go to the yes. So we call it incomplete dominancy. Yes. Then we go to the next question. The next question they're asking you, they're asking you that the point where two chromatids overlap during crossing over. How do you call the point where two chromatids overlap during crossing over? So in this case, the answer here is none. It's supposed to be none. The 2 points where the two chromatids overlap during crossing over, we call it chiasma. So the answer becomes A only. Yes. Then you go back and then see that the scientists who produced the law of segregation was discovered by, was proposed by Mendel. So in this case, there is nobody. So the answer becomes none. So it is none. It is A only. It is none. You see? Yes. 1.1.4. So we go to the next question, which is involving the discussion of 2 types of nucleic acids. We have the DNA and we have the RNA. So if you are talking about DNA and RNA, this is DNA, this is RNA. Why this is DNA? We said that is DNA because of one thing. Which one thing is because of the weak hydrogen bonds, while here there is no bonds. So identify the nucleic acid in the diagram 1. So we say that is DNA. Identify A. What is A? We said it's weak hydrogen bonds. Yes. And then he's saying that structure B, this is phosphate, phosphate phosphate group, or phosphate. Even if you say phosphate, you still get it right. Name the type of sugar found in nucleic acid represented in diagram 2. The sugar found there in diagram 2, because it is ribonucleic acid, therefore the sugar is ribose sugar. Yes. They are saying that which diagram 1 or 2 represent the nucleic acid used in the tracing of human ancestry. This one is we use what we call the mitochondrial DNA, therefore it's a DNA, therefore it's diagram 1. And then they're saying that paternity test, still we go for DNA test, therefore is still diagram 1. Yes. Then you go to question, the second and the next question. Next question is talking about the diagram below shows the karyotype. The karyotype of a gamete of an individual. So if you look at this karyotype, all of it, type of mutation is called chromosomal aberration or chromosomal mutation. So just say that that's chromosomal mutation. So the type of gamete which is formed here, the type of gamete, because now it has two gonosomes. So, and these two gonosomes are different. We can call it a hetero, because hetero means different. Heterogametic, gametic, heterogametic sex, or we can call it heterogamete. Yes, it's a heterogamete. Or yeah, that's the most scientific answer. Or it's a haploid gamete. Definitely is a haploid gamete. Yeah, and this will result in a person being a hermaphrodite. Yes, so in this case, so I'll call a heterogamete. So if you give a different answer, it could be correct depending on the scientific view of it. In this gamete, give the number of orosomes. Number of orosomes are this. Remember, they are single. Therefore, they are 22 orosomes. 22 orosomes. Not 22 pairs, it's just 22 chromosomes. Yes. And then gonosomes, if you look at them here, X, Y. Or, yes, it is X and Y, this one. And then if it fertilizes a normal gamete which has a Y, therefore is going to have XYY. So these are the two answers which we are looking for in this regard. You get a mark here, you get another mark in that regard. Yes. So 1.6, the diagram below shows, in this case, what are we looking at? We are looking at the diagram below shows the evolutionary relationship between the different species. So in this case, we are looking for different species. Identify. We talked about this phylogenetic tree, gave you all the answers, whatever they were asking you. It's basically in that distinction material book. So if you go down, yes, those questions here being even shown what we are discussing about when we are trying to predict the paper which is gonna come. So everything were given in that regard. So they're saying that, let's go and then see what are they saying. Phylogenetic tree. Yes, so we get a mark there, and they are saying genus that gave rise to Australopithecus africanus species. The genus which gave rise to Australopithecus at A. What genus is this? Obviously, is the first name. So we talk about the RDP Adipithe. Yes, that's the Adipithecus, which called the Ad. That's the genus which gave rise to that. Then they're saying that the letter that represents the most recent common ancestor for both Homo sapiens and Homo erectus. So you guys, this is what I was talking about, that we asked you this, what is the common ancestor of Homo sapien, Homo erectus. So we talked about all these things when we are discussing about the paper which is going to come, and then our prediction was 97% right. It's only I only saw just one small question where we couldn't spot it out. All right. Name two species that existed at the same time, meaning that they competed for resources between for Homorilla, and then we have the Pan. So basically, these are the two species which competed for resources in that regard. Then we go to the next question. They're saying that give two, give one example of fossil of Australopithecus africanus that was found in South Africa. In this regard, we are looking for the species. We said, I told you that always remember three species which were discovered in South Africa, that is Australopithecus africanus species. So in this case, we are supposed to say one of it, Taum, child. Yeah, number one, we can talk about Mrs. Place. Yeah, they say that this is a male, the female. Yeah, so if you wrote Miss Place, but it's better to say Mrs. Place because that's what it is still in the books. And then you can talk about the little foot. Yes, the little foot. So it's Taom Child, Mrs. Place, and then number three, little foot. They're just looking for just one. Yes, they are looking for just one. Guys, if you got 50 out of 50, congratulations. It's not 50%, it's 50 out of, so that the university or to be given a job or what. Yes, as long as you get decent, what you call the decent marks which is above your age. Yes, definitely. Yeah. But don't embarrass me. You get marks below your age. Guys, don't do that to me. Yes, it's like me spending the whole night trying to show you what's coming, and then you surprise me with such. All right, guys. All right, I know you did not do that to me. I know you guys, you got good marks. All right, let's go to question number 2. The diagram below represent the stage during synthesis of certain proteins. All right. So you have this, identify the organelle 3. This is a ribosome. It's a ribosome. Describe the role of molecule 1 in the stage of protein synthesis. So there they're just trying to ask you to describe the role of transferrin A. Basically, you're describing translation. Yeah, remember transferrin A carries the specific or the required amino acid to the ribosome. So it carries this amino acid and brings it to the ribosome. So it carries a required amino acid to the ribosome. So this is 2. So automatically, if this GGA, so it's gonna be CCU. So 2 is gonna be CCU. And then they're saying on the DNA molecule that code for histamine. DNA molecule, this is histamine, automatically is gonna be GTA. Yes, okay, let me show you how did we get the GTA. This is transferrin A converted to messenger, which is going to be CAU, and then convert it to DNA, which they are looking for, which is going to be G. A goes with the T, and then T goes with the A, and then gives you rise to that. So basically, that's how it's supposed to be done, because they said on DNA, and what is happening here is transferrin A. Therefore, you convert it into messenger RNA, then you convert it again into DNA. It is just a reverse. During synthesis of some proteins, there was a change in the amino acid sequence because alanine was replaced with the tryptophan. Alanine was replaced with the tryptophan. Explain the possible reason why this happened for this. Why was alanine replaced with the tryptophan with AA during the formation of messenger RNA? So that now if you have GG, it's supposed to be GG, yes. Now during the transcription, instead of G having CC, then it came to CAA. Eh? No. It came to CCUU. You understand? And then the transferrin A came to be GAA. And then instead of bringing the alanine, then it has to look for what code for GAA. GAA is tryptophan, so it's tryptophan which was brought instead of alanine. So in this case, where the problem came from is it came from during transcription, the two bases CC was replaced with UU. You see, was replaced with UU, and then the code became CUU. You understand? Yes, and then tryptophan was brought instead of alanine. So that's how we're supposed to explain. At least you're going to have to bring at least what? Here is what I told you, that emphasize this before, and then yes. So 6 marks, all the red thing means the marking point. Say that it's DNA double helix unwinds, yes, weak hydrogen bond break, unwinds, hydrogen bond break. So here there was another point which they include now to form two separate strands. Even if you don't write it, because you have too many points we can talk about, but if you wrote it, it's good, because that's the best. To form two separate strands, one strand act as a template to formation of messenger RNA using free-floating nucleotides from nucleoplasm. A messenger RNA is complementary to DNA, whereby A goes with U and C goes with G. And then yes, and then you're saying that the messenger RNA now has a coded information for protein synthesis, which moves out of the nuclear pore, out of the nucleus via the nuclear pore to the ribosome in the cytoplasm. So basically, to the next question, they're saying that the diagram below represents metaphase one of meiosis. Guys, this is a cell membrane. I told you that when the cell is under metaphase, anaphase, we don't have nucleus, nuclear membrane, we only have cell membrane. So the answer here is cell membrane. State one function of A. Function of A is to produce production of spindle fiber, which are used to hold the chromosomes in their position. Yes. Explain the contribution of metaphase one in natural selection. So in this case, they are looking at how is this metaphase one going to help us to bring to bring about variation. Remember, variation is the one which help us to survive in the what? In the environment. So how is that going to help us to bring about variation? Yes, that's the meaning. That's what they're trying to mean. Other side. What if this one is here? Automatically they will form different combination. So because they align along themselves along the equator randomly, we don't know which one is which side. So they create different different gametes. Yes, these gametes, they have different chromosomes, and then these different chromosomes, they us in forming different individual who are going to suit or who are going to adapt to the environment differently. That's why you don't resemble your sister, you don't resemble your brother, you don't resemble your mother. Why? Because of the different combination. Or you have one, two characters, you resemble your sister or your mother. Yes. And the others, they resemble their neighbors. I don't know how did you resemble your neighbor. Yeah. Don't say that you don't resemble. Haven't you ever gone somewhere and then someone say that, oh, when you resemble someone I know? How did you resemble that person? Anyway, maybe the father was a great man. Describe the diagram below shows 4 possible daughter cells from the at the end of this meiosis. All right, so draw the cell D, including the fourth chromosome. So here they want just to see the variations which are happening. When I was explaining to this, someone asking me yesterday that, sir, do we need to shade? I told them, yes, you need to shade because those variations, those shaded areas, they mean marks. So I know that some of you, this one gave you a headache. How did you find it? But it was just a very simple thing. Let me show you. You come here. So the way how these chromosomes move is that all these, they will go, the last one they will go in one cell, the middle one go in another cell, they go in another, because they separate by 2 2. You understand? So now if we say that black, white, black, black, so I will look for one black, one white, three blacks, so it means that's that cell. Then I continue, I go to black and white, black and white, black and white, black and white. So I'll look for the one which has black and white, black and white, black and white. So it's this one. It has black and white, black and white, black and white. Also this one, black and white, black and white, black and white, black and white. So you see that it's the next cell which has black and white. One, you see the carvings are for this one. Yes. This one, you see the carvings for this one. Then automatically the one which is missing is this. So it means that you are supposed to draw this cell again, and then you add a whitey one, and then it's supposed to show how straight it is, everything the way it is. That's how it's supposed to be done. And then maybe you put the labels, you label them, and then you put a caption, so that 1 mark for caption, 1 mark for label, and 1 mark for drawing a corrective, maybe diagram, whatever marks they will be allocated. So as long as you drew it the way I'm telling you. Yes, so guys, you see, meiosis tend to be simple but difficult when it, or complicated when it comes to the paper, but you just have to keep on revising and revising. Sex linked. Did you see anything here saying the sex linked? No. So no use X and Y. Explain why both individual 5 and 6 must be heterozygous for cystic fibrosis. Why are they supposed to be heterozygous? They're supposed to be heterozygous, these 5 and 6, because they produce a kids who are uh, having the disease, they're affected. So it means that one recessive allele came from 5 and one recessive allele came from 6. Therefore, they are supposed to be heterozygous for both the conditions. Now someone can say, why not small b? Small b is because they produce again individuals who are normal. Yes, they produce both individuals who are sick and or who are having the condition, and also those who don't have the condition. Therefore, they are supposed to be heterozygous. Use the letters B and C, okay, and then do the genetic cross to show the inheritance of fibrosis when heterozygous. Guys, this is just a simple thing. It's just a walk over for you guys. They just put this question so that you get those free marks. Yes. So they're saying that you have to say P1, obviously F1, that you tell us gonna be genotype is gonna be genotype. They said it is heterozygous, so it's capital B, small b. This one capital B, small b. Then you have meiosis. Meiosis is gonna take place. It's gonna be capital B, small b is gonna be capital B, small b, and then we call these ones gametes. Gametes. So you can draw a Punnett square method there, or you can just cross. So C, B with B, capital B, capital B, B with B, capital B, small b, B with B, capital B, small b, or small b, small b. So basically we call these ones offsprings, and then we call this one fertilization. For that one, fertilization. So in this case, that is how it's supposed to be done, and could be depending on whether you are whether you are they give a compulsory mark or not. So we come here, we give a mark for phenotype, give a mark for genotype, we give a mark for gametes, we give a mark for that, and then we give a mark for you telling us P1, F1, we give a mark for fertilization. Yes, you see all those marks. Maybe the compulsory mark was saying that you tell us the phenotypes. How many phenotypes? This is three normal, three normal, or without three normal, without fibrosis, and then one is affected with the fibrosis. And then when you go to the genotypes, you have one capital B, and then two capital B, small b, and then one small b, small b. So basically, that is it. That's how it's supposed to be done. So this question was just open for you guys to kill it and then get marks mahal. All right, let's go to the next question, that is 2.5. And then once we finish 2.5, we'll be able to have 100 marks. 100 marks out of 150, which is not a bad mark for you guys if you are a person who is looking for. Describe how genetic engineering of BTO was done. So in this case, they brought a gene of a plant which is resistant to those insects, and then they inserted it into this BT corn. Therefore, the BT corn started to be also resistant, and then they kept on reducing the amount of insecticide they are using. So they just crossed the gene for a gene which is from a plant which is resistant to those insects. Yes, remember insecticide, they kill the insects. Yes, so that's how it became to be more resistant. So describe the relationship between the land and the grow BT corn and amount of insecticide. Land to grow BT corn and amount of insecticide. So basically, they're asking you to describe. They're not asking you to explain. Explanation, you require a reason why. So in this case, they're just looking at as the number as the land size increases, yes, the amount of insecticide used decreases. Yes, as the land size increases, yes, land size used to grow BT corn, yes, increase the amount of insects. Two other plant characteristics that are genetically engineered to increase food security. Two other plant characteristics. They're looking for characteristics. You can talk about the size of fruit, the size of fruit. You can talk about the the shelf life. Yes, how long does it stay in there? You can talk about the sweetness of the fruit. You can talk about the juice. You can talk about, there are so many things you can look at just to increase the food security. Then lastly on this question is draw the bar graph to represent, let me see, the percentage land used between that here and here. So they are looking for only this. They are not only this and this. Therefore, year, since year is time, always is supposed to go on the x-axis. So the years is supposed to be here, 1995, 1995, 1995, 2000, what did I say, 2000, 2005, 2005, 2010. So like that. So maybe it's 0, this is 10, or 24, then the fifth one is 60. You see, yeah, remember when you're marking, don't forget guys that we're supposed to check here the size of this and the size of this are the same, size of this and the size of this are the same. Yes, also the space here is the same. Yes, the space is the same. Then now we give you the caption. Caption must have two the variables, this variable here and the variable here. If you look at here, this variable is saying that land used to grow BT corn in percentage. So you tell us that land used to grow BT corn, and then you put there in percentage. So we come and give you labeling the axis correct, and then you look at the scale. See the scale correct, then we also give you a mark for you having a correct scale. Then you come here, we give you maybe 2 marks for plotting properly. Then you give you a mark for correct type of the graph. then you give a mark for caption. You see, yes, that's how you accumulate marks. So you give 1, 2, 3, 4, 5, 6, yes, one maybe any other mark depending on their marking guideline. Yes, so you see that this question was also simple. You were able to get it. I would have kept myself on Thundereduc because really we try to give you everything you need. Yes, and I repeat, thank you guys, those who came back to me and said thank you, Mr. M.SAIDI, you really helped us. And also I'm saying thank you very much for not letting me down and keeping yourself on our channel. A certain country, 20 to 25,000 individuals were suffering from hemophilia between 2012 and 2020. Hemophilia is a genetic disorder caused by mutation. Pie chart below shows the percentage of individuals with the different levels of hemophilia in this country. All right, then you're saying that the percentage of individual, okay, cool. Explain the effect of mutation on individual. The mutation basically it causes the blood group not the blood not to clot. You understand? So you lose a lot of blood in a situation. Yes. Calculate the number of individuals who were moderately affected by hemophilia in the country. Show your working. In this case, because 1,250 people, so those are the people who were moderately affected by hemophilia. They're saying that explain why it would be expected that most of the individual affect hemophilia will be males. Basically, the reason why is like that because males contain one X. If a recessive allele appears on X, the individual is already affected by hemophilia. Why not female? Because females have 2X. If a recessive allele appears on one, it can be masked by a dominant allele and the individual is still normal. Yeah, that's why you see that it's very common in males. Why? Because males have one X chromosome. If a recessive allele appears on it, the individual is automatically sick. Yes. Then we go to the next question, which is 3.2. 3.2, what are they talking about? Here they talked about the insects on island with tree are likely to be wingless because they fly dangerously for them in area with a strong wind. Yes, you see, stoneflies have undergone a natural selection. Some have wings and some don't have wings depending on the area where they are coming from or they are found. The diagram below shows the stoneflies with and without the wings. So here is with the wings, here without wings. Scientists wanted to determine the relationship between the presence of trees and the wings on the stoneflies. The presence of tall tree, uh, tall tree, and wings on the flies. They selected 6 locations, or which 3 had tall tree plants, and then without tall tree plants. Using specialized net, yes, collected thousands of stonefly. This one is going to help us when you are talking about increasing the reliability. Yes, stoneflies, the location, the sample was labeled according to the area. Yes, these samples were all collected in the morning. Yes, you see the number of stoneflies wings and without wings at each location was counted and recorded. Presence or absence of tree, yes, on the island, of tree on the island, yes. And then this one is still presence or absence of the wings. So the wings will depend whether there is a presence of trees or there is absence of trees. Yeah, this is just a general question. State 2 factors that were kept constant. In this case, The factors which were kept constant is the collecting time. They were collecting in the morning, yes, collecting time, and also the method of collecting. Yes, they used the specialized nets. Yes, method of collecting, it was one, and the time of collecting was one. There are even others which you talked about and we have not talked about them. Yes, they also might be correct. State two ways in which the reliability of this investigation was ensured. Yes, was ensured. So standardized data collection, it is just a standard. The way how you are collecting data is the same. So that increases in reliability, and also replication of these individuals. So in this case, if you look at them, you'll find out that we used thousands of it would be it would have expected the more stoneflies with no wings in area without trees. We expect that because of the natural selection. How did natural selection? Those flies with wings could fly and then they're being blown away by strong wind. While these ones, you see, yes, by strong wind. While those ones without wings, they were holding themselves on the ground and they could not fly. Since they are holding themselves on the ground, therefore they could hide from predators, they survive, they produce large numbers, and they are able to increase their population. Yes, so basically those which had wings, they fly, blown away by by the wind, and also eaten by predators. Those without wings, they could not be blown away by the wind. They could hide from predators. They could survive. They will reproduce in large numbers. Basically, that is it in that regard, which is natural selection. While here, describe how Lamarck would have explained the evolution of stoneflies. When I was talking about Lamarck, only people know that the more used, the more frequently used the structure, the more becomes bigger and stronger. If you don't use it, becomes smaller and being applied. There was no variation among these stoneflies. All of them had wings. Yes, the stoneflies started to use the wings less frequently, and then the wings became smaller. You understand? And finally disappeared. This character of no wing was passed to the next generation. Then the next generation were born with no wings. That's it. That's how Lamarck explained. There was no variation among these 20 flies, meaning that all flies had wings, and then these flies started to use the wings less frequently. Then the wings became smaller and then disappeared. This character was passed to the next generation, and then all the flies were born without wings. So that's how you're supposed to explain that in that regard. Yeah, you see that the paper was not that bad, just some question required you to apply. Then they're saying that describe the process of speciation through geographical. This is another free mark, which I'm not going to take too much time in that regard. Explain this and emphasize this during the scope time or during the, yes, say that, yeah, this question will come. It is here. Okay, it's here. It's here. Punctuated. No, 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 no. That one we explained it. Yes, you see, even is highlighted nicely. It told you that 6 marks, 6 marks. It could be 7 marks. It doesn't exceed 7 marks, but it can go to 5 marks. So describe the process of speciation. This question is asking, describe the process of speciation by geographical isolation. So in this regard, what are you supposed to look at? You come here, you tell us that the population of a particular species were split into two by a geographical barrier, for example, a mountain. As a result of two parts of population cannot interbreed. So when you separated it, the two parts cannot interbreed. Like your home, the left side and the right side is not the same. You have different environmental conditions. Even in your class, there is a party spot you like to sit in when it's winter, like that part where maybe next to the window where the sun is coming, you want to bask the sun. Just a class, they don't have the same environmental conditions. What about when the geographical barrier is separating them? Definitely don't have the same environmental condition. Because of the geographical barrier, there is no gene flow, they could not cross each other. Typically different. Yes, they became genotypically and phenotypically different. I don't see people asking me questions. Ne? Maybe you got it right. Ne? Yes, if you have a question, let me know because I'm on live. Ne? It's not just talking. Ne? Not just talking. Ne? Yes, I'm on live. You understand? Yes. So if you what when you say as a result the two population became genotypical, you start with the word genotypically before you go to phenotypically because it's a genotype which determines the phenotype. Yeah. Yeah. Even if the two population mix later or again, they will not be able to interbreed. Yes, produce a fertile offspring. The word is fertile, not viable. So therefore, both or one or both population has new species. That's what they were looking for, so that you get your 6 marks in this regard. Mahala. I call them Mahala marks. Yes. Then we go to the next question, saying that list three. The diagram represents the skull of hominid. All right. These three similarities between for correct judgment. Name one species in the diagram that is prognanthus. In that regard, you are talking about the gorilla. It's gorilla which is prognanthus. Name one species. All right. Describe two structures that cause the species to become prognanthus or more prognanthus. First of all, we talk about the protruding jaws. Secondly, we talk about the flat face. The face is flat, therefore is the protruding jaw, and then the flat face. Those are the characters which we can also talk about the position of foramen magnum, but they said, yeah, two. All right. Hominid was a bipedal for most of its time or adult time. Yes. Explain how the structure of hominid skull would have assisted in bipedalism. Yes, the position of foramen magnum is a little bit forward, therefore it helped in connecting the spine vertically, and then is able to walk onto limb. Yes, the position of foramen magnum is a little bit forward, which helped to connect the spine vertically, and then she was able to walk onto limbs. So basically that's it. Describe the difference. One is short and wider, wider, and then this one is long and narrow, so that's if you go to our distinction material book, you'll find out that we highlighted this very well when it came to that. You see, it's short and wider, and then long and narrow. You see, long and narrow, and then this one less curved or C shape, and then this one more curved. Yeah, so everything was there, just that sometimes because of pressure, because of panicking, we panic and we end up forgetting our answers. Ish, that's a very bad thing. Ne? Yes. Explain why gorilla species have cranial ridges. They have cranial ridges. Cranial ridges are these ones. Cranial ridges are these ones. Yes, these things. Yes, these ridges. Yeah, it's not like human. They have cranial ridges for muscle attachment. Muscle attachment because gorilla they feed on raw food. Yes, so basically is for muscle attachment because it requires a strong is something. It looks like how do they call it? It looks like the trastramonia. It looks like a eggplant. Yes, it looks like eggplant, and it is very toxic. Yes, it used to kill soldiers. This thing is there. Flower open in evening and then release a powerful fragments which attract hawk moth. the Datura produce a high they produce highly addictive nectar, which highly addictive nectar, yes, which do what? Yeah. Which ensure that it ensures that the hawk's moth stay longer inside the flower and can only visit the Stramonia, the true flowers. So they're asking you explain two ways in which yes, explain two ways in which this tetra is ensure. Explain two ways is a key point. Basically, what they do is they are very important. They help in number one. They tend to open the flowers in the evening. Yes. So if they open the flowers in the evening, you see. So it means that other time of the day they are not able to do that. So they are a little bit isolated, and then they attract the moth. Yes. So basically, this is they have specific pollinator attraction, which is addictive. Yes, addictive. Yes, this one, which is addictive. So they try to produce a high addictive nectar, which ensure that the moth stay longer. Yes, you see. So opening at different specific time is basically issue. Yes. Step two, other mechanism by which by other mechanism that would ensure reproductive isolate can be basically special features on the plant which do not allow another plant to carry out cross pollination, or it could be isolated geographic